everyone, a very welcome to Vyas Edification Kota. So in the series of NCERT discussion today, we are going to discuss a new chapter from class 11, that is Plant Kingdom. Now, in this Plant Kingdom, we have total 12 questions in the NCERT and we are going to discuss each and every question in detail here. So let's begin, and we have a very first question, that is, what is the basis of classification of algae? Now question is asking on what basis the algae is being classified into different categories. So we know that algae is a member, or algae is a group of primitive plants included in the kingdom Plantae, and further algae is classified into 3 different classes, which is, we know that green algae, brown algae and red algae. The scientific name of these 3 is Chlorophyceae, Rhodophyceae and Phaeophyceae. Chlorophyceae for green algae, Rhodophyceae for red algae and Phaeophyceae for brown algae. Alright, these are the 3 classes in which the algae plants are divided into. Now, on what basis the algae are divided into these 3 categories? The main reason or the main basis of classification of algae was their pigments. The major pigments present in them was the main reason on which they classified into different categories. And also there is another reason, that is the presence of different food reserve material in them. All of these 3 categories of algae have different reserve food materials in them, as well as different major pigments in them. If we talk about the very first class, that is Chlorophyceae. Chlorophyceae is a group of algae, that is green algae. In green algae, if we talk about the major pigments, then all the plants included in Chlorophyceae have chlorophyll A pigment as well as chlorophyll B pigment. These 2 major pigments are present in the Chlorophyceae. Then if we talk about the Phaeophyceae, that means brown algae. The brown algae have pigments chlorophyll A and chlorophyll C as well as fucoxanthin. Alright, now if we talk about the last one, that is Rhodophyceae. Rhodophyceae have pigments chlorophyll A, chlorophyll D and phycoerythrin. So all these are the major pigments which are different in different categories and present in them, due to which, or on the basis of these major pigments, algae is being classified into 3 different categories, and these pigments are actually responsible for coloration or color present in the algae such as green, red and brown. Now let's talk about the food reserve material in different categories of algae. If it's starting with the Chlorophyceae or green algae, then the food reserve material in green algae or Chlorophyceae is just similar to the higher plants. That means Chlorophyceae algae have reserve food material as starch. If you talk about the Phaeophyceae or brown algae, then the food reserve material in brown algae is mannitol and laminarin. And the last one, if you talk about the Rhodophyceae, which is red algae, then red algae have food reserve material as Floridian starch, which is similar to the glycogen in structure. Now let's come towards the solution, written solution of this question. That is, if we talk about the basis of classification of algae, first we will write that the major pigments, present in algae is the main basis of classification and also the food reserve material, food reserve material present in them. Now we have three classes, we have three different categories of algae, which means that algae is being classified into three different categories such as green algae, brown algae and red algae. So let's write here chlorophyce, then we have brown algae, that means phaeophyce, and last one we have is red algae, that means rhodophyce. Alright, now we are going to write the major pigments of each of them and the food reserve material. So first I am going to write the major pigments of them. Then if we talk about the chlorophyce, we know that chlorophyce have chlorophyll A plus chlorophyll B. Now if we talk about the pigments of phaeophyce, that means brown algae, then we have chlorophyll A plus chlorophyll C plus fucoxanthin. Now in rhodophyce we have chlorophyll A. Chlorophyll A is common in each category of algae. Chlorophyll A, chlorophyll D plus phycoerythrin. Now let's talk about the food reserve material in them. The food reserve material in chlorophyce is starch. In phaeophyce it is laminarin plus mannitol, and in the last category of algae, that is rhodophyce, we have Floridian starch, which is similar to the structure of glycogen. In structure, it is similar to the glycogen. So here on the screen you can see the complete solution or answer of question number 1. Now let's talk about the second question, that is when and where does reduction division, that is meiotic division, takes place in the life cycle of a liverwort, a moss, a fern, a gymnosperm and an angiosperm. Now we are going to tell where actually the reduction division is taking place in each of these. First of all, we are going to see a liverwort life cycle, then a moss, a fern, gymnosperm, and finally we will move on to angiosperm. So to answer the question that where does and when does the reduction division or meiotic division taking place in the life cycle of each of them, we have to write or we have to know a life cycle of these plants. So first of all, we are going to discuss the life cycle of liverwort, which is a bryophyte. Now we know that the main plant body in a liverwort is gametophyte, which is a haploid plant body, that means N, a haploid plant body. So if we take an example of liverwort, we know that example of liverwort is Marchantia. Now Marchantia does not have true stem, root and leaves. The main plant body of liverwort or Marchantia is gametophyte, which is thalloid body. So I am going to make a thalloid body over here, which is having root-like structures, that is rhizoids, and this is a male gametophyte containing antheridium. Now another one we have is the female one, female gametophyte, that is female gametophyte containing female sex organ called as archegonium. So this is antheridium, and this is archegonium, in which the gametes are going to be formed. Let's suppose the egg cell is formed here and the male gametes are going to be formed in the antheridia, which are known as anthrozoids. Now after that, in female gametophyte containing archegonium, the egg will get fertilized with the male gamete, which will result into the formation of a zygote over here. In archegonium only, this is a zygote, which have ploidy 2n. Now we got 2n diploid zygote. This diploid zygote on gametophyte will develop and convert into embryo. This will convert into embryo. Now we will make, now what happens? This zygote will undergo mitotic divisions to form a sporophyte over here. We have sporophyte, which is also 2n. As we know that zygote is 2n, zygote is diploid, it will undergo mitotic division and form a sporophyte. Then after this sporophyte will undergo reductional division or meiotic division to form spores, and spores are going to be haploid in case of liverworts. These spores are made inside sporangium present on the sporophytic plant body. This is sporophytic plant body, which is made from the mitotic division of zygote. Zygote undergone mitotic divisions and formed sporophytic plant body. This sporophytic plant body contains sporangium, and inside sporangium meiotic division, reductional division occurs and result into the formation of haploid spores. These spores, each and every spore, give rise to a new plant body on germination. So this is a complete life cycle of a liverwort or Markantia, which is of course an example of liverwort only. This is a complete life cycle in which the two sex organs are present on two separate gametophytes, and they produce different gametes, on which fertilization they will give rise to a diploid zygote. This diploid zygote undergone mitotic division, which is not a reductional division, it is an equational division. Then on equational division, it will give rise to a sporophytic plant body of a liverwort, which is diploid in nature. Then sporangia present on the sporophytic plant body will undergo meiotic division or reductional division to form haploid spores. So now from this life cycle, we have clear idea that where and when does the reductional division or meiotic division is actually taking place in case of liverwort, as this is a life cycle of a liverwort. Now we have clear idea that, or we can write that when spores are made inside the sporangium of sporophyte plant body, sporophytic plant body, then the reductional division occurs. So this is the first we have done with that a liverwort. In liverwort, when and where does the reductional division takes place? When the spores are going to be formed inside the sporangium of a sporophytic plant body which is formed after mitotic divisions of zygote, then only the reductional division or meiotic division takes place. Now moving towards the next, that is moss. Let's discuss the life cycle of a moss first, by which we can get clear idea that in moss when and where the reductional division takes place. So we are taking a life cycle of a moss first. Now we know that moss is also a bryophyte, and the bryophyte, if we talk about the main plant body or the dominant phase of any of the bryophyte, it is always gametophyte. It is always gametophyte. So in case of moss, we know that a dominant phase of moss will be gametophyte, of course. Which is haploid plant body. Let's write about the liverwort. Liverwort at the time of spore formation. Now, if we discuss the life cycle of a moss, then we know that gametophyte is the main plant body or dominant phase in the life cycle of a moss. So we are starting with the gametophyte. We are starting the life cycle of a moss with gametophyte. Here we have gametophyte. Which is haploid in nature. Now what happens? We have two types of gametophyte, a male gametophyte and a female gametophyte. A male gametophyte and a female gametophyte. Let's suppose this is a female and this one is a male. Now, inside female gametophyte, egg cell will produced, and inside the male gametophyte, a male gamete anthrozoid will produced. Male gamete is produced. Now, on meeting of these two, what happens? A zygote will form. A zygote, which is 2n diploid, will form on the gametophyte plant body inside the flask-shaped female sex organ, that is archegonium. This zygote is being formed inside the archegonium only. This is archegonium. Archegonium, which is present on the gametophytic plant body of a moss, and suppose this is your gametophytic plant body. Now inside the archegonium, zygote has been formed. This zygote is diploid in nature. Now after this zygote will convert into embryo. This will develop into embryo and this will undergo mitotic division. So from here we can conclude that a zygote will undergo mitotic divisions. Mitotic division, which is an equational division, and convert into develops a sporophyte, and sporophyte will develop on gametophyte only. A sporophyte which is developed from the zygote, it will retain on the gametophytic plant body only. So here suppose we have gametophytic plant body. This is gametophytic plant body. And on gametophytic plant body, inside the archegonium only, a sporophyte will develop like this. A sporophyte will develop like this, and this sporophyte have three components. That is, first one you say is capsule, second one is seta, and the last one is foot. Alright, So this sporophyte, this is a sporophyte which is 2n, this is sporophyte and 2n, which is formed from the mitotic divisions of zygote. Now, inside this sporophytic plant body, which is dependent on gametophyte and retaining on the gametophytic plant body of a moss, it will have sporangia inside which spores are going to be formed. So sporophytic plant body, let's suppose we are taking here sporophytic plant body. Inside sporophytic plant body, meiotic divisions will occur. Meiotic divisions will occur and it will give rise to haploid spores here. Haploid spores. So, in a moss also, at the time of formation of spores inside the sporangium of a sporophyte which is retained on the gametophyte, the reductional division takes place. We have concluded that in a moss, at the time of spore formation, alright. Now let's talk about the fern, which is third one. Now what happens in fern? As we know that fern is a pteridophyte. Now what is the main plant body in case of ferns? What is the main plant body of a fern? As it is a pteridophyte, it is a vascular plant, and if we talk about the main plant body, then fern have main plant body, or the dominant phase of fern is sporophyte. And sporophyte have ploidy level as 2n. That means the plant body, the main plant body of a fern is sporophyte, and that is diploid in nature. Now, in the life cycle of a fern, what happens? Let's talk about, this is sporophyte. Two N. Sporophyte will contain sporangia. Alright, in sporangia, this is the sporangia. They will have sporangia inside which the haploid spores are going to be formed. But as we know that sporangia is 2n, diploid, as it is a part of a sporophytic plant body of a fern. Then sporangia having diploid structure, the cells present inside the sporangia will undergo which kind of division to form haploid spores, and spores are always haploid. So which kind of division is required here to form haploid spores from sporangia? It will reductional division, that is meiosis or meiotic division or reductional division. So after the formation of haploid spores, these spores will germinate into gametophyte, and these gametophyte will develop archegonium and antheridium over them, inside which the gametes are going to be formed. Now these gametes will fertilize with each other and form a diploid structure again, that is zygote, which is 2n. This zygote undergoes mitotic divisions. This zygote will not go meiotic division, it will undergo mitotic division and equational division to form a sporophytic plant body again. So this is a life cycle of a fern, in which we have seen that in the beginning of a life cycle of a fern, which is a sporophytic plant body, the main plant body of a fern is a sporophytic plant body which is containing sporangia, a modified sporophyll, inside which the cells are present. These cells undergoes meiotic division or reductional division to form haploid spores over here. So we have seen that meiotic division takes place at the formation of a spores in the beginning of the cycle only. And why it is in the beginning of a cycle? Because if we are talking about the fern, then fern is a pteridophyte, and pteridophytes have their main plant body or a dominant plant body as sporophyte, which is containing sporangia. And of course, for the formation of spores, the sporangia, the cells present in the sporangia will undergo meiotic division. So a fern will also have reductional division at the beginning of the cycle of life, at the beginning of a life cycle in them, plus at the time of the formation of a haploid spore. Now let's come towards the next, that is gymnosperm. Now in case of gymnosperm, we know that gymnosperm is a modern plant. It is an advanced type of plant. In this also we have sporophyte as their main plant body. The sporophyte is a main plant body in them. Now we are starting with the life cycle of gymnosperm. Here we have sporophyte. In case of sporophyte, which is 2 and of course diploid in nature, they contain female cone as well as male cone. Alright, female cone and male cone. Female cone is also known as seed cone and male cone is also known as pollen cone. Now what happens? Inside female cone, they have megasporangia or macrosporangia. And if you talk about the male cone, they have microsporangia. Now, these sporangias will give rise to spores, haploid spores. Now, this sporangia, megasporangia, have special cells, megaspore mother cells, will give rise to haploid spores, egg cell and pollen cells. This will give rise to male spores, or we can call it microspores, and here we can call it megaspores. Now we know that spores are always haploid and sporangia. Sporangia is diploid 2n in structure, as it is a part of cone and cone is a part of a sporophytic plant body, main plant body in a gymnosperm. Then the cells present in the megasporangia and microsporangia will undergo which kind of division to form haploid spores? Of course, meiotic division. Now these spores are going to give rise to gametophyte over here. Gametophyte, and gametophyte will develop archegonium and antheridium, in which the gametes are going to be formed, and then zygote will develop. Then this zygote will undergo mitotic divisions and give rise to a sporophytic plant body again. So this is the complete life cycle of a gymnospermic plant, and what we have seen in this life cycle of a gymnosperm plant, that at the time of formation of a spore only, the reductional division takes place in them. But when the megasporangia, or we call them at the time of formation of spores, and where if we talk about the where, then in the megasporangia and microsporangia, the reductional division takes place, which are the part of female and male cones present on the main dominant plant body, that is sporophyte of a sperm. So we have concluded then when and where the reductional division takes place in case of gymnosperms. Now we are left with the last one, that is angiosperm. Let's talk about the life cycle of angiosperms. Angiosperms are the most advanced plants, and the main plant body or the dominant plant body of angiosperms is always sporophyte. The sporophytic plant body is diploid in nature, and it contains a special reproductive structure that is flower reproductive structure. That is flower. Flower can be unisexual, flower can be bisexual depending upon the species. If I talk about the unisexual flower, that means a flower contains either only male reproductive part or female reproductive part. And if you talk about the bisexual one, then bisexual flower contains both type of reproductive structures, that is male reproductive structure as well as female reproductive structure. So here as example we are taking the bisexual flower, as flower is the main reproductive origin, main reproductive place where the all type of divisions, either it may be reductional division or mitotic division, are going to take place and are forming different structures. So we are taking flower which is bisexual, these are the anthers, stamen of course containing anthers and filaments, this is pistil. Now this flower is bisexual. What happens inside this? This is anther, this is filament. Inside anther there are present microsporangeas. So what happens inside microsporangeas? Or microsporangium, there are 4 microsporangeas. So microsporangia which are diploid in nature, as they are part of the plant only, they are part of the flower only. Flower is also a diploid structure as it is present on the sporophytic plant body of an angiosperm. Then microsporangia will have megaspore mother cell or microspore mother cell or pollen mother cell. So MMC we can write microspore mother cell or PMC pollen mother cell. These microspore mother cells or PMC will undergo meiotic division here. Meiotic division here, that means reductional division, and they are forming spores like this. They are forming spores which are going to develop into pollen grain. These are pollen grains which are male gametophyte. These pollen grains in case of angiosperms are male gametophytes. So these are gametophytes. These gametophyte pollen grains of angiosperm containing 2 type of cells in pollen grain, that is vegetative cell and generative cell. Generative cell will give rise to 2 male gamete. Alright, 2 male gamete. So from here gamete will be received. And in case of the female reproductive structure, this is ovary. This swollen part at the base of carpel is known as ovary. Inside ovary we know that there is a presence of ovule which is attached to the placenta through a funicle, a small stalk-like structure, funicle. Inside the ovary of a carpel, in case of angiosperm, we know that inside the ovary we have ovule which is a megasporangium. Ovule is the megasporangium in case of angiosperms. That ovule is attached to the placenta with the help of funicle. So this is your ovule, and ovule is your megasporangium. Megasporangium, which is also diploid, and this is ovule. ovule. Ovule is your megasporangium. Now, inside megasporangium or ovule, we have special kind of cells or tissue, that is nucleus tissue. Sorry, nucleus tissue. And these nucleus tissues have already MMC, that is megaspore mother cell, which will undergone meiotic divisions again, meiotic divisions to form megaspore tetrad, and among this tetrad, 3 will get degenerate, one will remain functional, one will remain functional and give rise to embryo, and thus it will forming a new plant body of angiosperm. So this is a complete life cycle or an overview of a life cycle of an angiosperm. So from this overview, we can conclude that in case of angiosperms or in angiospermic plants, the reductional division is taking place inside the microsporangia and megasporangia only, but megasporangia and microsporangia are present inside of flower, which is a reproductive structure in case of angiosperms. So when and where the reductional division is taking place in case of angiosperms? At the time of formation of spores, at the time of formation of male and female gametophyte. So this is a pollen grain, which is a male gametophyte and female gametophyte here. So at the time of formation of female and male gametophyte, the reductional division takes place in case of angiosperms. And if we talk about where does it takes place, then inside flower or in a flower, there are two kind of reproductive structures are present, that is male reproductive structure and female reproductive structure, inside which these reductional division or meiotic divisions are taking place. So we have already seen and we have already discussed each and every kind of plant, that is liverwort, moss, fern, gymnosperms and angiosperms, and we have seen when and where does the reductional division takes place. So if we conclude and if we write a summary of this, then if we talk about the first one, that is liverwort, then at the time of formation of spores, and if we talk about the moss, then the same thing will goes for moss at the time of formation of spores only, and for fern also the same thing will goes for fern. For gymnosperm, the same thing will goes for gymnosperm and angiosperm. If we talk about angiosperm, then at the time of formation of gametophyte, alright, at the time of formation of gametophyte, the reductional division takes place. And if we talk about the place or where does the reductional division takes place, then we know that in liverworts, sporangia is the site. In moss, it is obviously sporangia, which are present on sporophyte only. These two are present on sporophyte. These two are present on sporophytic plant body. And in case of sperms, obviously the same will happen. Sporangia, which is present on sporophyte, and this is also sporangias, but they are present on cones. And in case of angiosperm, there is speciality or special features. If I write for angiosperm, then in angiosperm, these reductional divisions are occurring in sporangia only, but these sporangias are a part of flower. These are occurring in a flower. So this is a complete solution of question number 2. Let's move on towards the next question, that is question number 3, and this question is asking us to name 3 groups of plants that bear archegonia, and also we have asked to briefly describe the life cycle of one of them. Alright, now we have to tell which of the following groups of plants actually bears archegonia. First of all, what is archegonia? Archegonia are the female sex organ which is present on the plant body. Alright, now archegonia is the female sex organ inside which the female gametes or egg cells are being produced. First of all, this is the definition of archegonia. Now, which of the following or which of the groups of plants actually have archegonia in their plant body? So in the plant kingdom we have algae, bryophytes, pteridophytes, gymnosperms and angiosperms. 5 divisions we have in the kingdom plantae, among which we have only 3 phylums in which archegonias are found. So let's see which of these following. So the 3 groups of plants that bears or contains archegonia are 1. Bryophytes. Alright, bryophytes are the group of plant which contain archegonias as their female sex organ. Along with this we have pteridophytes. This group of plant also contains archegonia, and the last one we have is gymnosperms. Gymnosperm being advanced plants, but they are the plants or group of plants which contain archegonia as their female sex organ. So all these 3 group of plants are the groups that contain archegonia. And what is archegonia? It is a female sex organ which produces female gamete or egg cell. Now we are going to discuss the life cycle, one of them. Then when we are going to discuss the life cycle of a pteridophyte, alright, a fern. So if we are talking about the life cycle of fern or a pteridophyte, we know that in case of pteridophytes, the main plant body, the dominant plant body or the dominant phase in the life cycle of a pteridophyte is actually sporophyte. Alright, the plant body, the main plant body or dominant phase in pteridophyte is sporophyte. Now these sporophyte have special modified leaves known as sporophylls. On these sporophylls there is the presence of sporangias. Now these sporangias are present in the sporophyll, on the sporophyll, and these sporophylls are the part of sporophytes. Sporophytes are diploid in nature. They are always diploid in nature. As the sporophyte or sporophytic plant body of a pteridophyte is diploid in nature, the sporophylls present on the sporophytic plant body are also be diploid in nature, and the sporangias present on the sporophyll are also diploid in nature. Now what happens? These sporangias are containing spore mother cells. Those spore mother cells are also diploid in nature. Then these spore mother cells undergo meiotic division or reductional division to form haploid spores. These sporangias have diploid spore mother cells. These diploid spore mother cells undergoes meiotic division or reductional division to form haploid spores. These haploid spores give rise or germinate to form a new individual plant and that individual new plant which is arised from the germination of a spore is actually gametophyte. This gametophyte in case of pteridophyte is known as prothallus and is haploid in nature. This haploid gametophyte or prothallus when develops or get matures, it develop two type of sex organs on its own body, that is male sex organ and the female sex organ. The male sex organ is antheridia and the female sex organ is archegonia. The antheridia will make male gamete or anthrozoids and the archegonia being female reproductive organ will make or produce egg cell or female gamete. Now, with the help of water, anthrozoids or male gamete will reach to the archegonium and fertilize the egg present in the archegonium. After the fertilization, the zygote will develop inside the archegonium only. Now, after the development of zygote or formation of zygote, zygote will undergo mitotic divisions to form again a sporophytic plant body. So, this is the life cycle of a fern or pteridophyte which we are going to make here on the screen. So, let's make a life cycle here. So, if we are talking about the fern's life cycle, we know that the sporophytic plant body. Sporophytic plant body this is sporophyte, which is 2N, and we are talking about the fern. Alright. Now, this sporophytic plant body of a fern is containing sporophylls like this. These are the modified leaves, sporophylls. Inside which the spores are present. The spores are present. Alright. In these spores, we have special kind of spore mother cells. Let's suppose these are the spore mother cells. Spore mother cells, which are diploid of course, as they are present inside the sporangia, and sporangia are present on the sporophylls, which is a part of a sporophytic plant body. Now, these spore mother cells will undergo meiotic division or reductional division to form haploid spores. Haploid spores. Now, each of these spore will undergo germination, and on germination they will produce a haploid plant body which is known as gametophyte or prothallus. In case of pteridophytes, prothallus. These spores on germination give rise to a thalloid body, which is a gametophyte haploid plant body of a pteridophyte, and it is known as prothallus. This prothallus will develop antheridium and archegonium, inside which the egg cell and male gametes are formed. Now, with the help of water, these antheridium will go to the archegonium and fertilize it, and they form zygote. So, this is zygote, which is diploid in nature. Now, after the formation of zygote, this zygote will undergo mitotic divisions to form again a new plant body, which is sporophytic plant body and is diploid in nature. So, it will undergo mitotic division to form sporophytic plant body. So this is a life cycle of a fern or a pteridophyte, and we have three groups, that is bryophyte, pteridophyte and angiosperms. These are the three group of plants which contain archegonium in them, and archegonium is a female reproductive structure or a female reproductive organ inside which the eggs or female gametes are going to be formed. So this is a complete solution of question number 3. Now let's discuss question number 4, that is mention the ploidy of the following protonimal cells of a moss. Then we have primary endosperm, nucleus and dicots. Then we have to mention the ploidy of leaf cell of a moss, and then prothallus cell of a fern. Then gamma cell in Markentia, meristem cell of monocot, ovum of a liverwort and zygote of a fern. So let's discuss the ploidy level of first thing, that is protonimal cell of a moss. Now what do you mean by protonimal cell of a moss? Now we know that moss is actually a bryophyte. Alright, now in case of bryophyte, the main plant body, the dominant phase, is actually a gametophyte. And if we are talking about the protonimal cell, then it is the earliest stage of development of gametophyte in case of mosses. And protonimal cell is a thread-like structure or a thread-like chain which is green filamentous and is a gametophyte. So protonimal stage or protonimal cell is the earliest stage of development of gametophyte of a moss. And as we know that any gametophytic plant body of any of the group of plants is actually haploid in nature, then all the cells present in a gametophytic plant body are haploid in nature. Thus we can conclude that protonimal, first of all if we write answer, then protonimal cell is a thread or chain of cell that is and is gametophyte and is gametophyte. Now, as we know that gametophytes of any plant body is actually haploid in nature, then we can conclude then the protonimal cell, the protonimal cell of moss or bryophyte is haploid in nature. Alright, next we have is primary endosperm nucleus in dicots. Now we know that dicot and monocots are two groups under the phylum or division Angiospermi in the kingdom Plantae. We know that angiosperms are the most advanced plants and they are also known as flowering plants. Angiosperms are further classified into two categories on the basis of presence of cotyledons in them. That is, if any group of plant is having only one cotyledon, then they are considered as monocotyledonous plants or simply monocots. And on the other hand, if any group of plant is having two cotyledons in them, then they are considered as dicotyledonous plants or simply dicots. Now, if we have asked the ploidy level of primary endosperm nucleus in dicots, we know that in gymnosperms, both in dicots and monocots, in whole gymnosperm group of plant, there is a reproductive structure called as flowers. Flowers can be bisexual or unisexual. In case of both unisexual or bisexual flowers, we know that there is a female reproductive structure present known as carpel or pistil. The carpel is composed of three parts, that is stigma, style and ovary. Inside ovary, which is the base or swollen part of carpel or female reproductive organ, inside ovary there is a cavity known as ovarian cavity. Inside ovarian cavity there is Megasporangium or ovule is present in them. The ovule present in the ovarian cavity of a carpel or pistil in case of Angiosperms is actually attached to the placenta with the help of a funicle or a stalk known as funicle. Now, as we know that the ovule is considered as Megasporangium in case of Angiosperms. Inside the Megasporangium or ovule there is a special tissue present known as nucellus, and a special kind of cell is also present there, that is megaspore mother cells. These megaspore mother cells give rise to megaspore tetrad. These megaspore mother cells undergoing meiotic divisions will develop or give rise to megaspore tetrads. That means megaspores are being formed. Now, tetrad means the formation of 4 spores, 4 megaspores from megaspore mother cell. Megaspore mother cells are diploid in nature and the spores formed from the megaspore mother cells are now haploid in nature. Among all 4 megaspores, among all 4 haploid megaspores, 3 of them gets degenerate, and 1 remains as functional. That one functional megaspore will give rise to embryo sac. That one remain functional megaspore which is haploid will give rise to an embryo sac. An embryo sac will have 7-celled 8-nucleated stage. This embryo sac have 7-celled 8-nucleated stage like this. You already are familiar with the structure of embryo sac. So this is your embryo sac which is formed from single haploid megaspore. Now, this embryo sac have 7-celled 8-nucleated stage. On one end it have 3 cells like this which are known as antipodal cells. On the another end it have egg apparatus made up of 3 another cells, that is known as egg apparatus, and the middle one in egg apparatus is known as egg cell, and the side 2 cells, the 2 cells are actually known as synergids. Now, this egg cell is containing egg or female gamete inside it, and now in the center of this embryo sac we have 2 nuclei present in center. Alright, 2 nuclei are present in the center which is known as central cell in the embryo sac. These 2 nuclei are known as polar nuclei. These polar nuclei fuses together to form a secondary nucleus, Let's suppose they are fused. Let's suppose they have fused like this and they formed secondary nucleus. Now the ploidy level of this nucleus will be 2n. Alright. Now what happens after the formation of male gamete inside anther, or we can say in male reproductive structure? The pollen grains or male gametes are being transferred to the embryo sac. The male gametes are being transferred to the embryo sac to get fertilized with the egg cell present in the embryo sac in egg apparatus. Let's suppose this is your male gamete. Male gamete has came and 2 male gametes we have. Alright, 2 male gametes are present in a pollen grain. One male gamete is fuses with the egg cell to form a zygote. The remaining 1 male gamete gets fuses with this diploid nuclei or nucleus present in the central cell of embryo sac. Now let's suppose this male gamete fuses with the secondary nucleus. 2n plus n or nucleus of male gamete, when fuses with each other they form triple n condition. We know that this is the secondary nucleus containing 2n. fuses with the haploid nucleus of sperm, it forms triple N condition or triploid condition. Triploid condition. This formed structure from the fusion of this diploid or secondary nucleus and haploid nucleus of male gamete is known as primary endosperm nucleus, or in short we can call it PEN. So now it is clear that the ploidy level of PEN or primary endosperm nucleus will be 3N, triploid. It is triploid in condition. Now, if we talk about the leaf cell, next is leaf cell of a moss. We have to find the ploidy level of a leaf of a moss. As we know that in case of moss, which is a group of plant under bryophyte phylum, so we know that moss have dominant face or dominant plant body as gametophyte. Gametophytic plant body means haploid plant body. That means the whole body containing each and every cell with only half set of chromosome in them or haploid nucleus in them. So if any gametophytic plant body of a moss is haploid in nature, then each and every cell of that gametophytic plant body or of a moss is haploid in nature. So leaf cell of a moss is also haploid in nature as it is a part or it is present on the gametophytic plant body of a moss. So it is simply haploid in nature. Next, come towards the ploidy level of a prothallus of a fern. Now, prothallus of a fern means it is a gametophytic plant body which is formed from the gametophyte, alright, which is formed from the gametes. Now, let's discuss. Now, if we talk about fern, we know that in case of fern, the male plant body or the dominant plant body is sporophyte. This sporophyte, the sporophytic plant body of a fern will give rise to spores, will give rise to haploid spores. Now these haploid spores, each of these haploid spores will germinate and form gametophytic plant body. This gametophytic plant body is also known as prothallus. Now, as we know that gametophytic plant body is haploid in nature and is known as prothallus, so gametophytic plant body of fern or prothallus of a fern is actually haploid in nature. So ploidy level of prothallus cell of a fern would be haploid. This will also be haploid. Now, next we have gamma cell in Marchantia. Now, what is Marchantia? Marchantia is a liverwort, is an example of liverwort. Now, gamma cell is actually part of asexual reproduction. Gamma cell is a part of gamma cup. Gamma cups are the structure in which the gamma buds are being produced in Marchantia, through which Marchantia or liverworts can reproduce asexually. So these gamma cups are also haploid in nature, as in liverworts. The main plant body or the dominant plant body is also a gametophyte. So being gametophyte as their main plant body or dominant plant body, the gamma cups present on the main plant body gametophyte are haploid in nature. Thus, each and every cell, including gamma cell, are haploid in nature. So these are also haploid in nature. So let's write here gamma cell. Gamma cells are also haploid in nature. All right. Protonema cell are also haploid in nature of moss. If we talk about the endosperm nucleus, primary endosperm nucleus, that is triploid in nature. Leaf cell of a moss is haploid in nature. Prothallus of fern is also haploid in nature. Then the gamma cell of Marchantia is also haploid in nature because the dominant phase, the dominant plant body in them is gametophyte. Then come towards the next ploidy level of meristem cell of monocot. Now meristem cell of monocot. Let's come towards the monocot and dicot. As we know that angiosperms are the most advanced plants, and the angiosperms have their dominant phase or dominant plant body as sporophyte, which is diploid in nature. Now we know that the sporophytic plant body is diploid in nature, and each and every cell, the leaf cell, the stem cell, the flower cell, each and every cell present on the sporophytic plant body is also being diploid in nature. So if we talk about the meristem cells are the totipotent special cells which will give rise to different parts or structures of the plant and helps the elongation and development of plant. So meristem cells are the totipotent cells present on the sporophytic plant body of angiosperms only. Now as we know that the main plant body of monocot as well as dicot plants in case of angiosperms is sporophyte in nature, and the sporophytic plant body are diploid in nature. Now each and every cell present on the sporophyte of monocot and dicot are diploid. So hence we can conclude that meristem cells of monocot are also diploid in nature. So meristem cells of monocot as well as dicot are diploid in nature, because they are the part of sporophytic plant body, and sporophytic plant body is diploid in nature. Then second last we have is ovum of a liverwort. Ovum means egg cell or female gamete, and they are talking about a liverwort. We know that liverwort are having their main plant body as gametophyte, and the gametophytic plant body is haploid in nature. If gametophytic plant body is haploid in nature and is producing sex organs, female sex organ as well as male sex organ, then the respective gametes, female gamete as well as the male gamete formed inside the female reproductive structure and male reproductive structure would be haploid always, as the main plant body, the dominant plant body in case of liverworts is haploid in nature. Thus we can say the ovum or egg cell or female gamete produced on the female gametophyte of a liverwort would also be haploid. So ovum of liverwort is haploid in nature, because it is being produced inside female reproductive organ, that is archegonium, and archegonium is a part or a structure present on the female gametophyte which is haploid in nature. Then the last is zygote of a fern. Zygotes are always diploid. Even in lower plants, if zygote is forming during the sexual cycle, sexual reproduction, then zygotes are always diploid. Without exception, zygotes are always diploid in nature. So we have completed the solution of question 4, and this is a complete answer. Let's come towards the next question, that is write a note on economic importance of algae and gymnosperms. So we have to discuss the economic importance of algae and gymnosperms. Here we will be writing the importance of algae, and here we will be writing the economic importance of gymnosperms. So first talk about the economic importance of algae. algae alone fixes half of the carbon dioxide present on earth by the process of photosynthesis. Algae have huge or a diverse economic uses. First is it alone fixes half of the carbon dioxide present on earth by the process of photosynthesis, and also algae is known as primary producers of aquatic habitats. Now if we talk about the food source of algae, yes, algae have many species of algae such as Porphyra, Sargassum, Laminaria. These are edible algae. Along with these, we have Spirulina and Chlorella. These two are extremely rich in protein content and are used as food supplement. If you talk about another thing, commercial purpose of algae, then you are familiar with a substance known as agar. Agar is a substance used in the manufacturing of jellies, ice creams, etc. Now, this agar substance is obtained from Gracilaria and Gelidium, which are the two species of red algae. Next, carrageenan. Carrageenan is a substance which is used as emulsifier in chocolate and toothpaste. This carrageenan product or substance is obtained from red algae, of course. Then algae have other medicinal purposes also. Corallin. Corallin is a algae which helps in the treatment of worm infection. Now, along with that, we have some functions and some economic important functions of gymnosperms also. If we talk about the economic importance of gymnosperms, they are used or they have a huge economic importance in the construction purpose. So in construction purpose, what are they, the gymnosperms are being used, how they are being used? Some species of gymnosperms such as Pinus and Cedar, they are good manufacturers of softwood, and the softwood obtained from these gymnosperms are used for the construction purposes, for furniture as well. If you talk about the next purpose of gymnosperm or economic importance of gymnosperm, there is a species in gymnosperm known as Texas, which produces Texon, and it is used as anti-cancerous drug. An anti-cancerous drug known as Texol is obtained from a species of gymnosperm that is known as Texas, and also many species of gymnosperms known as Ephedra produces ephedrine, which is a drug and is useful in the treatment of respiratory diseases such as asthma and bronchitis. Also, we have some food resources of food economic importance of gymnosperms. Some species of Pinus produces seed known as Chilgoza, which are edible in nature. Now, raisins are the substance obtained from plants, and they are used as water repellent, or they're used in sealing and waterproof paints. Raisins are used in the making or manufacturing of sealing and waterproof paints, and these raisins are obtained from the gymnosperms. A very famous or common raisin known as turpentine is obtained from the gymnosperm, and which of the species from gymnosperm? A turpentine, which is a common raisin, is obtained from the species of Pinus from gymnosperm. So let's write about the economic importance of algae. First, we'll write these are the organisms which fixes half carbon dioxide on earth, all right, and they are known as primary producers in in aquatic habitat, in aquatic habitat. Many species of algae such as Porphyra, Sargassum, Laminaria are edible. Along with these we have Spirulina and Chlorella are also edible in nature and they are rich in protein content. If we talk about the medicinal purpose, then we have Corallina and is used in treating worm infections. Last but not least, if we talk about agar is a substance which is used in the manufacturing of ice creams and jellies and obtained from Gracilaria and Gelidium, which are the members of red algae only. Now let's come towards the economic importance of Gymnosperm. One we have already written, that is resin, which is used in the manufacturing of sealing products and waterproof paints, is obtained from the many species of Gymnosperm, that is Pinus. And if we talk about the edible or food sources from Gymnosperm, then a species of Pinus produces seed which is known as Chilgoza and is edible in nature. Also it is used in construction purposes, because they produces soft wood. If we talk about the medicinal purpose, then an anti-cancerous drug, Taxol, is obtained from Texas. And if we talk about the next thing, that is ephedra. Ephedra is a species of Gymnosperm and it produces ephedrine, which is used in the treatment of respiratory diseases such as bronchitis and asthma. So these are the economic importance of algae and Gymnosperms, and we have completed with the solution of question number 5. Let's come towards the next question, that is question number 6, and is asking both Gymnosperms and Angiosperms bear seeds, then why are they classified into separate categories? So we know that Gymnosperms and Angiosperms are the advanced plants, and both these group of plants are included in the kingdom Plantae, and being advanced they also bear seeds. Alright, Gymnospermic plants as well as Angiospermic plants, both kind of plants or both the group of plants are having seeds on them, but the basic difference is that in Gymnosperm the seed is not enclosed by any fruit wall or fruit, and in Angiosperm the seed is not naked, the seed is enclosed by a fruit wall or a fruit. So this is the basic difference on the basis of which they are classified into different or separate categories. So answer would be like this, that both Gymnosperms and Angiosperms bear seeds, but the basic difference is Gymnosperms have naked seeds, have naked seeds. Now what is naked seeds? We already know that gymnosperm is a word which means naked seed. That is, naked seed means the seed, that is the seed is not enclosed, within the fruit, or is not enclosed by any fruit wall, whereas the angiosperm plants, angiospermic seeds are enclosed. They are enclosed by a fruit wall or fruit, and this is the basis, this is the reason they are classified separately. They are classified separately, and this having enclosure of seed by fruit wall or fruit makes angiospermic plants the most advanced plants on earth. And this is the basic difference between gymnospermic plants and angiospermic plants, that in gymnosperms seeds are naked seeds, that means they are not enclosed by any fruit wall or fruit, and in case of angiosperms, the angiospermic plants or angiospermic seeds are enclosed within fruit or fruit wall. This is the basic difference, and this is the criteria by which they are classified into separate categories. So this is the complete solution or answer of question number 6. Let's come towards the next question, that is question number 7. What is heterospory? Briefly comment on its significance and also give 2 examples. So let's understand what is heterospory first of all. So heterospory is a phenomena in which 2 different types of spores are being produced by the same plant only. A plant body, same plant body is going to produce 2 different kind of spores on its own. Now this phenomena is known as heterospory. Now what happens? These 2 heterospory or these 2 heterospores, different type of spores, are being produced on the same plant, but they differ in size. The one is smaller in size and the another one is larger in size. The small sized spores are known as microspores and the large spores are known as megaspores. These microspores are male spores and going to give rise to male gametophyte. The larger one, the large megaspores, are female spores and are going to give rise to female gametophyte, inside which the male gamete and female gametes are going to be formed. After the formation of gamete, the male gamete will flow with the help of water and reaches to the female reproductive organ where the egg cell or female gamete is present and fertilizes with it. After the fertilization, the zygote will develop, but the zygote will develop inside the female reproductive organ only. That reproductive organ, female reproductive organ, will retain on the gametophytic plant body only. And this is the main step, this is the main thing which is actually participating in the evolution of seed habit. This retention of megaspore, this retention of zygote on female gametophyte ensures the better development of zygote or embryo, which actually participating or a significance, importance in the seed evolution. This retention of megaspore on the female gametophyte only is the main step in the seed evolution, as it is a precursor of seed habitat. And if you talk about the example of heterospory, then we have two examples such as Selaginella and Salvinia. For the first time, heterospory is being introduced in the Selaginella and Salvinia of pteridophytes only. So, if we want to write the answer of this, then heterospory, let's write heterospory is a phenomena in which different kind of spores are produced. As the name suggesting itself, heterospory means different kind of spores are being produced by the same plant only, by the same plant. Now what happens? First of all, this is the phenomena. What is spory? Heterospory. Heterospory is a phenomena by which two different kind of spores are being produced by the same plant only. Now what happens? Suppose this is a plant. This is a plant and this produces different kind of spores. Plant is having sporophyll, which is a modified leaf. The lower base of sporophyll having large megasporangia and smaller ones on the top of the sporophyll. These are microsporangia. These are microsporangia, and the larger one sporangias are known as megasporangia. Megasporangia, inside which the spores are being produced. If we talk about the microsporangia, then it will produce microspores, and in megasporangia, megaspores are being produced, megaspores, which will give rise to male gametophyte, and female gametophyte, These male and female gametophyte will develop their reproductive structures or organs respectively, inside which the gametes are being produced. Let's suppose if we're talking about the male gametophyte will develop antheridium and female gametophyte will develop archegonium. Inside antheridium, the male gametes or anthrozoids will form, and inside archegonium, the egg cell or female gamete is going to be formed. After the formation of gamete, let's suppose this is your gametophyte. Gametophyte is having archegonium, this is egg cell, and here we have gamete, male gamete. These male gametes are coming towards the egg cell and fertilizes with it and forming a zygote. Alright. Now after the formation, this zygote and this structure retain on the gametophytic plant body only. This is retained on the gametophytic plant body only, and this retention of these structures on the gametophytic plant body ensures this retention of the gametophytic plant body ensures the proper development of zygote, and this is actually important. This is important for evolution of seed, as we know that if zygote is developing inside the megasporangia and megasporangia is present on the sporophyll, archegonia is present on the sporophyll, this is the archegonia, and this retention of zygote, this retention of zygote on the megaspore and on the gametophyte is actually main step in the evolution of seed habit, as it is precursor of seed habit. Alright, so this is the significance of heterospory, and if you talk about the example, then Salvinia and Selaginella. These are the two examples of heterospory. So this is the complete solution of question number 7. Let's come towards the next question. Question number 8. Explain briefly the following terms with the suitable examples. Alright. Now first term we have to explain is Protonyma. Protonyma is actually a green leafy filamentous autotrophic free living gametophytic stage in the life cycle of a moss. Alright, Protonyma is a stage in the life cycle of a moss which is green, autotrophic, free living, and it is produced from the spore germination, which is going to give rise to new gametophytic plant. And if we want to write, then Protonyma is stage, we are talking about the first, it is a stage of life cycle in mosses. So example would be moss, of course, and this is green autotrophic this is green autotrophic free living gametophytic stage, which is going to give rise to a whole new plant as gametophyte. An example we have is mosses. Let's talk about the antheridium. What is antheridium? Antheridium is a male sex organ inside which the male gametes or anthrozoids are going to be formed. So what about antheridium? Antheridium are the male sex organs inside which the male gametes are formed. And if we talk about the example of antheridium can be found in bryophytes. They can be found in bryophytes, pteridophytes, alright, and also in gymnosperms. Now next let's come towards the archegonium. Archegonium is the female reproductive structure. Archegonium is the female reproductive or sex organ inside which the female gamete or egg cell is produced. And also if we talk about the example of archegonium, then archegonium is found in pteridophytes, bryophytes and gymnosperms as well. Then these are the same examples for archegonium. And if we talk about the next thing, that is diplontic, then diplontic is actually a type of life cycle. This is a type of life cycle in which the main plant body is sporophyte. In diplontic type of life cycle, which is exhibited by angiosperms and gymnosperms, the main plant body is diplontic. The main plant body is diploid in nature, and these diploid plant bodies are going to form haploid gametes by the process of meiosis. All right, and this diplontic life cycle is exhibited by angiosperms and gymnosperms, which are the example of this. Next, talk about the sporophyll. Now, what is sporophyll? If you talk about the sporophylls, sporophylls are the modified leaves. These are the modified leaves. These are the modified leaves containing sporangias, inside which the spores are going to be formed. Sporophylls can be microsporophylls as well as can be megasporophylls. Microsporophylls have microsporangias, inside which the microspores are going to be formed, which are male spores, and in case of megasporophylls, which are larger and have larger sporangias known as megasporangias. Inside megasporangias, the megaspores or the female spores are going to be formed. Sporophylls can be found in pteridophytes as well. Now let's come towards the next and last term, that is isogamy. Isogamy is a type of sexual reproduction. Isogamy is a type of sexual reproduction in which the fusion of two similar gametes occur. Similar gametes in size and as well as in function, they come together and fuses with each other to form a zygote. This type of reproduction, this type of sexual reproduction in which the similar type of gametes in size as well as in functions fuses with each other to form a zygote is known as isogamy. And isogamy can be found in chlamydomonas, Eulothrix and Ectocarpus. So these are the examples and these are the meanings or explanations of each of the term given on the screen, and this is a complete solution of question number 8. So let's talk about the next question, that is question number 9. And in question number 9, basically we have 4 parts. As you can see on the screen, here we have the first part of question number 9, which is differentiate between the following, and we have to differentiate between red algae and brown algae. So the answer goes like this: red algae are grouped under the class Rhodophyceae. When we are comparing the red algae and brown algae, we will differentiate and we will write all the characteristics of red algae here and the characteristics of brown algae here. First of all, we are just discussing the characters or differences between the red algae and brown algae. Now, as red algae are actually grouped under the class Rhodophyceae, if we talk about the food, reserve food in them, in red algae, it is Floridian starch, which is similar to the glycogen in structure. If we talk about the chlorophyll pigments present in red algae or Rhodophyceae, then they contain chlorophyll A, chlorophyll D and phycoerythrin. Then if we talk about the flagella are absent in the members of red algae or Rhodophyceae. Whereas if we talk about the brown algae, then brown algae are grouped under the class Phaeophyceae. The pigments, the main pigments present in the brown algae or the members of Phaeophyceae are chlorophyll A, chlorophyll C and fucoxanthin. If we talk about the reserve food material of brown algae or Phaeophyceae, then it would be mannitol and laminarin, and flagella are found in them. So let's write about the differences between red algae and brown algae here. Now, we have to differentiate between red algae and brown algae. In the left column, I am writing the red algae, and here brown algae. So the first point would be red algae are grouped under the class Rhodophyceae and the first point of brown algae is that brown algae are grouped under the class Phaeophyceae. Phaeophyceae. The next thing which we are talking about the red algae is the presence of food material. The presence of reserve food material in them is they contain Floridian starch. Floridian starch which is similar to the glycogen in structure. To the glycogen in structure. Alright, the Floridian starch is the reserve food material in Rhodophyceae or in red algae. If we talk about the next, that is brown algae or Phaeophyceae, then they contain laminarin, mannitol as their reserve food material. Next thing which we are going to talk about them is, or the next difference we are going to talk about them is the main pigment, the major pigment, chlorophyll pigment present in them. So they contain, we are talking about the red algae or Rhodophyceae, they contain chlorophyll A, chlorophyll D and phycoerythrin. Alright, if we are talking about the pigments present in brown algae or Phaeophyceae, they contain chlorophyll A. Chlorophyll A is common in all of the three classes of algae, Rhodophyceae, Chlorophyceae and Phaeophyceae. So chlorophyll A, they contain chlorophyll A, chlorophyll C and also they contain fucoxanthin. If we talk about the flagella, then flagella are absent in the members of Rhodophyceae. Flagella is not found in them. Flagella are absent in the member of Rhodophyceae or in red algae. And if we talk about the algae, the brown algae, then flagella are present. Flagella are present in them. Alright, so these are the basic differences between red algae and brown algae. Now let's talk about the second part of 9th question, and here we have to differentiate between liverworts and mosses. Now as we know that both liverwort and mosses belongs to the bryophytes. Bryophytes are the primitive plants and they have thyloid body. Their body of liverworts and mosses included in the bryophyte do not differentiate. We cannot clearly differentiate the body of liverworts as well as mosses into true stem, roots and leaves. Hence we can call them as thyloid bodied organism. Alright. Now both of these liverworts and mosses belongs to the bryophyte, but we have to differentiate between both of them as they actually have some differences between them. So let's talk about the differences between liverworts and mosses. So if we're talking about the liverworts, then liverworts do not contain true roots. There is no presence of true roots found in liverworts. Instead of true roots, they have root-like structures present in them. Root-like structures called rhizoids, but rhizoids are present in mosses as well. Then what is the basic difference between liverworts and mosses rhizoids? The basic difference between rhizoids of liverworts and mosses is liverworts have unicellular rhizoids, only unicellular rhizoids, and in case of mosses, mosses have multicellular rhizoids. If we talk about the liverworts, liverworts have scales very often are present in them. In case of mosses, the scales are completely absent in mosses. Next, if we talk about the body, then the body of liverwort is thalloid, and in case of mosses, the body is foliage and it is actually laterally branched. Now, if we talk about the next thing in the liverworts, then the gamma cups inside which the gamma buds are being produced, which is a means of asexual reproduction. The gamma cups are present in liverworts, and the gamma cups in case of mosses are completely absent. Alright, now we have to write the differences between liverworts and mosses. So let's write the differences. Here we are going to talk about the differences between liverworts. These are liverworts, and here we have mosses. Alright, now what is the basic difference? What is the first difference of liverwort? That the rhizoids which are not true roots, they are just root-like structures present in them, and rhizoids are unicellular in case of liverworts, and in case of mosses, the rhizoids are actually multicellular. Alright. Next thing, if we talk about the body of them, then the body is thalloid. The body of liverwort is thalloid, is completely thalloid body they have. Alright, that means the body of liverwort is unable to differentiate it between true roots, stems and leaves. Alright, and if we talk about the next thing, that is mosses, then mosses have actually, the body is foliage, the body is foliage with lateral branching. Alright. Next thing, if we talk about the liverwort, then the scales are oftenly present in them, and scales are completely absent. In case of mosses, scales are completely absent in them in case of mosses. Next thing, if we talk about the reproductive structure, asexual reproductive structure, that is gamma cup, inside which the gamma buds are being produced for asexual reproduction. So gamma cups are present in them, and the gamma cups are absent in case of mosses. And last but not least, if we talk about the sporophyte of liverwort, Then the sporophyte of liverwort have very less photosynthetic feature. Sporophyte of liverworts have very less photosynthetic pigment. All right. Next, if we talk about the mosses, then mosses have sporophyte, the sporophyte of mosses. The sporophyte of mosses have high photosynthetic pigment or large photosynthetic pigments. Alright, so this is the complete answer of question number 9th, second part. Next is the third part of question number 9, that is differentiate between the following, and we have to differentiate between homosporous and heterosporous pteridophytes. Now, what is the difference between heterosporous and homosporous pteridophyte? We know that two types of pteridophytes are found everywhere. That is, first if we talk about homosporous, this is homosporous, and this is heterosporous. Now what happens in homosporous pteridophyte? Only one type of spore, only one type of spores produced. Only one type of spores are produced on the sporophylls. Alright, which will give rise to bisexual gametophyte. And in case of heterosporous pteridophyte, there is presence of sporophyll, and sporophyll have two types of sporangias. One is microsporangia, and another one is megasporangia. It is megasporangia, inside which the microspores and megaspores. Microspores are being formed inside the microsporangia, which are male reproductive structure and male spores, and inside megasporangia, which is a female sporangia. Inside megasporangia, the female spores are going to be formed. Now, these two different spores we will get, female and male separate spores are produced inside microsporangia and megasporangia. Then these female megaspores are going to form female gametophyte, which will develop archegonium, which is the female reproductive organ, is developed on the female gametophyte, and the male microspore produced male gametophyte, which is going to develop antheridium on it, and inside antheridium the male gametes are going to form. After the formation of gametes, the male gamete is going to transfer to the female gametophyte, inside which the archegonium is present containing egg cell, and it will fertilize the egg cell. So this is the difference between homosporous and heterosporous. Homosporous pteridophytes are those pteridophytes inside which the homosporous pteridophytes have sporophylls, inside which only one type of sporangias are present, and in these sporangias there are sporogenous mother tissues or sporogenous tissues present, which will undergo meiosis and give rise to haploid spores. But only one type of spores are being produced inside the Homosporous Pteridophytes, and all these spores will germinate to give rise to gametophyte, and those gametophytes, all the gametophytes produced from these kind of spores are bisexual. That means the whole gametophyte is containing both the type of reproductive organs, male reproductive organ as well as the female reproductive organ, antheridium and archegonium. And in case of Heterosporous Teridophyte, what happens in them? They have different sporophylls, they have different sporangias of course, and if we talk about the small sporangia, in the sporophyll, the small sporangia contains sporogenous tissues which undergoes meiotic division to form microspores. Microspores are male spores, and in the large sporangias which are present at the base or lower surface of the sporophyll, they contain sporogenous tissues which undergoes meiotic division to form megaspores or macrospores, which are female spores. Now in Heterosporous Pteridophytes, there is formation of two different type of spores occurs, microspores and megaspores. Microspores are male spores, megaspores are female spores. Microspores are going to give rise to male gametophyte on germination, whereas the megaspore give rise to the female gametophyte on germination. Once the gametophyte is formed, each of the gametophyte, the male gametophyte and female gametophyte, will develop reproductive organs on them. The male reproductive organ antheridium is going to be developed on male gametophyte, whereas the female reproductive organ, that is archegonium, is going to be formed on female gametophyte. So this is the basic difference between Homosporous Pteridophyte and Heterosporous Pteridophyte, that in Homosporous Pteridophyte and Heterosporous Pteridophyte, spores are different in them and spores are similar in them. The gametophyte, basically the gametophyte formed in Homosporous Pteridophyte are bisexual, and in them always the gametophyte is unisexual. Always the gametophyte is unisexual, because there are two different type of gametes, there are two different types of spores present, and two different types of spores are going to give rise to different types of gametophytes on germination. So this is the complete solution of third part of 9th question. Now let's come towards the last part of question number 9, that we have to differentiate between syngamy and triple fusion. Now you are all familiar with the reproductive cycle or sexual cycle of angiospermic plants. Angiospermic plants are the most advanced plants in kingdom Plantae. We know that, and in angiosperm the speciality feature or the special feature is the presence of flower. The flower is actually reproductive structure in angiospermic plants, inside which the male and female reproductive structures are present. That flower, which contains both the type of reproductive structures, are known as bisexual flower. And in flower, where the only one, either it may be male reproductive structure or female reproductive structure is present, then the flower is said to be unisexual. Now after the formation of gametes in their particular reproductive organs, we know that in a sexual reproduction we need two types of gametes, that is male gamete and female gamete. So male gamete is going to be formed in male reproductive structure, and in case of angiospermic plants, the male reproductive structure is stamen, constituting of anther and filament. The male gametes or male gametophyte pollen grain is going to be formed inside the microsporangia present in anther. And in case of female gamete, that is egg cell, that egg cell or female gamete is going to be formed in carpel or pistil. Where in carpel or pistil? Inside the ovary section or inside ovary where the ovule is present. All of these things or gametic formation happens inside the ovule. After the formation of these different types of gametes, the gametes have to fuse with each other to form a diploid structure known as zygote. This is the basic rule of sexual reproduction. Now we know that after the formation of two different types of gamete, there is fusion comes. Fusion of male gamete and female gamete. So the process of fusion of male gamete and female gamete is known as fertilization, and in case of plants, in case of this thing, we call the fertilization as syngamy. So I am writing here that syngamy is actually the process of fusion of male and female gamete, which result into the formation of, we know that, which result into the formation of a diploid structure known as zygote. Formation of zygote, which is a diploid structure through which the whole organism is going to arise. This process of fusion of two different types of gamete is known as syngamy in case of angiospermic plants. Now let's come towards the triple fusion. Triple fusion. Now we know that in angiospermic plants, the ovule containing megaspore mother cells will give rise to megaspore tetrad. In those 4 cells, 3 of them gets degenerate, 1 remains functional. The remaining functional megaspore is going to give rise to embryo sac, inside which we find 7-celled 8-nucleated structure or stage. Then what happens in embryo sac? Embryo sac is containing 3 antipodal cells and egg apparatus. In egg apparatus, there is presence of 3 cells. One middle cell is known as egg cell and the side 2 cells are known as synergids. The central cell have 2 polar nuclei which fuses with each other to form secondary nucleus. Now, after the formation of gametes in their respective reproductive organs, the formation of male gamete and female gamete in their respective organs, they have to fuse with them. So, for the fusion of gametes, the pollen grain, which is a male gametophyte, is being shedded on the stigma of a carpel. Now, the pollen tube is going to be formed. The pollen tube will enter into the embryo sac through synergids, where the male gametes, the 2 male gametes are going to be transferred into it. Now, among the 2 male gametes, one is going to be fused with the egg cell, and the process of fusion of one male gamete and egg cell is termed as syngamy, which is going to give rise to a diploid structure known as zygote. The remaining male gamete is going to fuse with the secondary nucleus present in the embryo sac. We know that we have an embryo sac like this, inside which the 3 antipodal cells are present. Here we have egg apparatus. Here the zygote is being formed. Now, the zygote is being formed after the syngamy process. Here is the secondary nucleus. Secondary nucleus have 2 aneuploidy because 2 nuclei were present initially in the embryo sac central cell, which fused together to form secondary nucleus. The other male gamete is present over here. The other male gamete will go into the secondary nucleus and fuses with it, forming a triploid structure known as primary endosperm nucleus, and this primary endosperm nucleus is triploid in nature. So what is triple fusion? Triple fusion is this. Triple fusion is this. Now if we want to write the definition of triple fusion, then one male gamete when fuses with the secondary nucleus, present in the central cell of embryo sac, then this type of fusion is known as triple fusion, and as a result we will get PEN. The result, triple fusion will result into the formation of a PEN, which is triploid in nature. That means primary endosperm nucleus. So this is the complete solution of question number 9, and we have completed all the parts of this question number 9. Now let's talk about the next question, that is question number 10, and in the question number 10 we have to differentiate or distinguish between monocots from dicots. Now both monocots and dicots belong to the phylum or division Angiospermae, which is the most advanced plants on earth. But we have to differentiate between monocots and dicots. So if we talk about the monocots, then monocots are known as monocots because of the presence of one cotyledon in their embryo. Whereas if we talk about the dicots, then dicot embryo have two cotyledons. Alright. In case of monocots, the pollen grains of monocots have one pore generally, and in case of dicots, the pollen grain have 3 or more germinal pores in them or furrows in them. If we talk about the flower in monocots, flower of monocots may be trimerous. Alright. If we talk about the dicot plants, then dicot plants have tetramerous or pentamerous flowers. If we talk about the venation of leaves in monocot plants, then it would be parallel venation, and in case of dicot plants, the venation is reticulate. If we talk about the root of monocot plants, they are adventitious, and if we talk about the root of dicots, they have arise from radicle. If we talk about the vascular bundle of monocots, the monocot stems have vascular bundles, and the vascular bundles in the stems of monocots are present in the scattered form, whereas in dicots, the vascular bundles in the stem of dicot plants are present in a ring manner. Alright, and last but not least, the monocots have no secondary growth, and if in case of dicots, the secondary growth is present in them. Now let's write the differences between monocot and dicot. Here we have monocot, and here we have dicots. So let's write about the very first thing, that is, these are monocots because the embryo have only one cotyledon, and in case of dicots, the embryo have two cotyledons, That is why the name given to these kind of plants is dicotyledon plants, dicotyledonous plants or dicots, and the name of these plants have given as monocots or monocotyledonous plants. Next, if we talk about next thing, then the flower in them is trimerous flower. That means the flower parts are present in them in the multiples of 3, and in case of monocots, dicots, The flowers are present in the form of tetramerous flowers, or pentamerous flowers. Alright, if we talk about the furrow in pollen grain, then the furrow in pollen grain is generally 1, and in case of dicots, the furrow in pollen grain is generally 3 or more. Now, the venation, leaf venation is parallel in them, and in case of dicots, the leaf venation is reticulate. It is reticulate, network like structure. Then the vascular bundles are present in the scattered form, whereas in dicots, the vascular bundles are present in ring manner or ring form. No secondary growth is found in case of monocots. No secondary growth. And in case of dicots, secondary growth is present. So this is the complete solution of question number 10, that monocots have only one cotyledon in their embryo. They have two. The flower is trimerous, that means the floral parts are in the multiples of 3 in them, and the floral parts of dicots are present in the multiples of 4 or in multiples of 5. They have parallel venation, they have reticulate venation. The furrow in pollen grain is 1. They have 3 or more furrows in their pollen grain. The vascular bundles are scattered form. They are arranged in a ring manner. There is no secondary growth found in monocots, whereas secondary growth is found or can be seen in the dicot plants. Now let's talk about the second last question of this chapter, question number 11, and we have to match the column 1 options with column 2. So column 1 have A. Chlamydomonas, B. Cycas, C. Selaginella, and D. Sphagnum. So in the column 1 we have given the species, we have given the plants, and in column 2 we have given the groups. Now we have to match the column 1 with column 2. So this is a Chlamydomonas, and we know that Chlamydomonas is a member of Algae. So this is the member of Algae. So we will write like here, answer A. Chlamydomonas. Chlamydomonas is actually belongs to Algae. This belongs to Algae. Next we have is B, and in B we have Cycas. Now Cycas is the member, Cycas is the species of Gymnosperms. This is gymnospermic plant. Alright, so Cycas is Gymnosperms. Then we are left with Selaginella and Sphagnum. So Selaginella is a member of Teridophyte. And Sphagnum is a member of moss. So C. Selaginella. Selaginella is the member of Teridophyte. And the last one we have is Sphagnum. Sphagnum, which is a member of moss or bryophyte. So we have completed with the solution of question number 11. Now let's talk about the last question of this session, that is describe the important characteristics of gymnosperms. Now gymnosperm is the group of plant included in the kingdom Plantae, and these are the advanced plants. Alright, the most advanced plants are Angiosperms included in the kingdom Plantae. Gymnospermic plants are those plants, or I may say the term gymnosperm to these plants is given because of the presence of naked seed in them, or we can call these plants are naked seeded plant. Why is it so? Because the seed are produced in them, the seeds are produced in these plants, but the seed is not enclosed by any ovarian wall or ovary, or we can say fruit. The seed is produced in them, but the seed is not enclosed by any fruit. Alright. Now, if we talk about the body of plant of gymnosperms, it can vary from shrubs, it can vary from small trees to tall trees and shrubs. The redwood sequoia is the tallest tree in the world, which belongs to the gymnosperms. If we talk about the root, then root system is taproot system present in all the plants of gymnosperms, as well as the coralloid roots can be seen in the roots of cycas, which is present in the form of symbiotic relationship and are associated with the nitrogen fixing cyanobacteria such as Anabaena and Nostoc. If we talk about the leaves, then leaves can be simple as in case of pinus, and it can be compound or pinnately compound leaves are present in case of cycas. Now, if we talk about the flower, they lack flower. None of the tree, none of the plant included in the gymnosperm have flowers. Pollination can occur in them by the help of wind and insects. Now, if we talk about the sporophyll, megasporophyll and microsporophyll, they are heterosporous. First of all, they are heterosporous, they are not homosporous. They are heterosporous, that means they produce two different type of spores in them, the microspores and megaspores. If we talk about the sporophylls, which are the modified leaves containing sporangia inside which the spores are going to be formed, they have different kind of sporophylls, megasporophylls and microsporophylls. These microsporophylls and megasporophylls aggregate themselves and form a compact structure known as cones. There are two different kind of cones found in them, female cone as well as male cones. Alright, so these are the general features, these are the important characteristic features of gymnospermic plants. So let's write about these. The first is they are known as naked seeded plants. Naked seeded plants. Why they are known as naked seeded plants? Because the seed produced in them is naked and is not enclosed by any fruit wall. Second thing is the body or the plant body of them can vary from small to tall trees and shrubs. One of the plant belonging to the category of gymnosperm, that is redwood sequoia. Redwood sequoia is the tallest tree in the world. So you have to remember this, that redwood sequoia is the tree belonging to the gymnospermic plants and is the tallest tree in the world. Then if we talk about next thing, that is root system. root system is found in them is taproot system. Taproot system is found in them. In case of cycas, coralloid root is present. Coralloid root is present. Coralloid root is present, which is associated with nitrogen fixing cyanobacteria, that is Anabaena and nostoc. Now let's talk about the leaves. Leaves can be simple. In case of Pinus and Cedrus, and it can be pinnately compound. It can be pinnately compound in case of cycas. Then they do not possess flower. Flower is absent in them. Pollination in them can occur by wind, mostly by wind. Pollination can occur in them by wind. Then they are heterosporous. That means two different type of spores are being produced in them. Different type of spores are found in them, which are produced in microsporophylls and megasporophylls respectively, and hence we can conclude that they have female and male cones. They have male and female cones. So these all are the general and important characters of gymnosperms, and we are done with the answer or solution of question number 10. And with this, we have completed all the questions given in your NCERT of this chapter. So this is all for today. Thank you and have a nice day.